All right, so here's where we left things off last time, but obviously this is getting kind of messy. So I'm going to redraw this whole thing for you without all the extra little bits. All right, we've got the basic sequence, animals, eumetazoans, bilaterian, protostomial common ancestor. Then up from the first animal, we've got the phylum Porifera. Then you've got the radiata, the phylum Cnidaria coming off from the first eumetazoan. The bilaterian. From the bilaterian, we go up to the deuterostomial common ancestor, which gives us both the phylum Echinodermata as well as the phylum Chordata. And then from the protostomial common ancestor, we've got the Lophotrochozoan common ancestor as well as the Ecdysozoan common ancestor. And the Lophotrochozoan gives rise to the phylum Annelida, or the annelids, the mollusks, as well as the flatworms, the phylum Platyhelminthes. And the ecdysozoan, he'll give us the arthropods as well as the nematodes or roundworms. See the improvement? Now, as I recall, we were talking about the split right here, where the eumetazoan was splitting into a diploblastic cnidarian as opposed to a triploblastic bilaterian. And the difference between those two was basically the presence of mesoderm, a muscle-like material that allowed bilaterians to move around more effectively than they could in the absence of mesoderm. Now, the other thing that's really obvious is a difference between the radially symmetrical radiatans and the bilaterally symmetrical bilaterians is the way that they differ in their body symmetry. Put things relatively technically, but also very simply. Radially symmetrical organisms have a principal axis of body symmetry. Let's make this one go from the top to the bottom. This will be the primary axis of body polarity. And on one end, we'll have the oral end, and on the other end, we'll have the aboral end. Aboral just basically means not the oral. But if we think about the body plan as something like a sea anemone, this is exactly what we have. So here's your sea anemone, drawn many times in the past. And yeah, we've got an oral end, the mouth side, and the other side that has the aboral. There's basically no secondary axis that's going to make one side of the animal any different from the other side. You can't say there's a right and a left, or a front or a back. Pretty much everything is the same all around this entire axis, right? All the way around. This is radially symmetrical. So a pizza would be something that had radial symmetry. A slice of an orange would have radial symmetry. The tire of a bicycle would have radial symmetry. And these are all things you can pretty much explain to a third grader. Okay. Now, in the case of bilateral symmetry, you have two axes of polarity, two axes of body polarity. So I'm going to try to do this in a three-dimensional way. Let's say we've got a principal body axis that goes from the front to the back. Okay, so this is going to be our primary axis. This will be the anterior. This will be the posterior. That's the primary axis of symmetry. But then you've got an orthogonal axis. You've got an axis that goes in the other direction. It's going to be the up-down one, right? We call it the secondary axis, or sometimes we call it the directive axis. And it's going to go from dorsal to ventral. If we have two axes of polarity, this creates basically a whole plane of symmetry. So this plane is kind of like this two axes determine a plane. This is going to correspond to the center line of, say, a fish. And the fish is going to have a right side and a left side. So if I draw the fish in here, say here's the mouth. And so you've got a right side of the fish on this side, and the other side is over there. And you've got the left pectoral fin and the anal fin, which is along the midline. So basically you've got a right side of the fish and the left side of the fish. This would be the right on this side. This would be the left on this side. Okay. So yeah, bilaterally symmetrical or bilateral, meaning they have two sides. And bilateral symmetry is characteristic of these animals like fishes, of snails, of humans, and pretty much all of the bilateria have this type of symmetry. Now, the one piece I want to add to this is that we've got two different kinds of symmetry, radial and bilateral, and these are largely associated with two kinds of ecologies, in particular how the organisms are moving around. In the case of sessile or planktonic organisms, we tend to get a fairly large predominance of radial symmetry. And this kind of makes sense because if you're sessile or planktonic, there's no real direction that you're moving. Okay, it's kind of like you're pretty much at the mercy of the currents if you're planktonic, or if you're sessile on the bottom, you're 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 going to be getting food from all different directions. If you're a sea anemone, you don't know if the food is going to be coming from this side or from this side or from this side. There's no predictability, and there's no reason for you to designate a front and a back, a right and a left. Okay. On the other hand, with bilateral organisms, uh, these are these are animals that either crawl or swim. Because if you're swimming around or crawling around along the bottom, you know what direction that the food is going to be coming. The food is going to be in front of you. You move in the direction from posterior towards the anther. You move in the direction towards your nose, and so obviously the food is going to be coming in from this side. And so it's actually a good idea for you to have an anterior and a posterior. And because we're moving in a particular direction, it's also a nice thing for us to designate a top and a bottom. This is where the dorsal and ventral, the secondary axis, is coming in. And so the rule of thumb that I like to give you is that the bilateral organisms tend to be ones that move around, whereas the radially symmetrical organisms tend to be ones that don't actually have any physical movement. Now, the thing about this is that it makes perfect sense. A mesoderm goes along with this type of ecology because it's the mesoderm-derived tissues like muscle and bone that facilitate the act of moving through the world, crawling and swimming. In the case of the radiata, like the Cnidaria, the animals that have radial symmetry, they don't have mesoderm, and so they could deal pretty well with either sessile or planktonic ways of life. They're not going to be doing a whole lot of moving, right? So, getting back to our earlier picture, you could say that the evolution of bilateral symmetry, along with the third germ layer, might be actually associated with an ecological shift from being sessile, planktonic in the ancestral state towards being uh, fully locomotory and actually depending on crawling or swimming and moving around their world in order to make their living. Right? Now, the other thing I want to point out here is that all of the events that I've discussed, all of these origins, like the origin of the animal, the first animal, the origin of the first eumetazoan, even the origin of the very first bilaterian, this is all very deeply placed within the Precambrian. Not too deeply. It's all Proterozoic. In fact, the Neoproterozoic, the most recent time within the Proterozoic. But none of this is documented in the fossil record. We don't have any idea of what the very first bilaterian looked like. Okay, there's a big question mark with regard to that first bilaterian. What did it look like? Even how long ago it existed. We suspect that it must have been very, very small. We expect that it must have been very, very simple because we've got two extremely different kinds of body plans coming from that. And so a bilaterian ancestor, a protostomial, deuterostomial ancestor, must have been extremely unspecialized to allow the deuterostomial lineage, the one that gives rise to us as well as starfish, to be developing in such a different way compared to the protostomial lineage. Okay. And by the time we actually get to the beginning of the Phanerozoic, we have all 8 of these more complex phyla in place. They appear for the very first time, completely distinguished from all the other animal phyla during the Cambrian period, the very first time of the Phanerozoic. The presumption, of course, is that they must have diverged from each other. The deuterostomial common ancestor must be within the Proterozoic. The Lophotrochozoan, the Ecdysozoan, and certainly the protostomial ancestor are before the start of the Cambrian, and the bilaterian common ancestor must be even earlier than that. Okay, so we're going to be spending a little bit of time talking about what we might be able to infer about that first bilaterian, how long ago it existed, what it might have looked like, what might have its genome looked like based on the information that we can get from the evidence provided by our 8 modern, well-characterized animal phyla. And I want to point out that this is nowhere near a complete inventory of the animal phyla that have ever existed on the earth. There's something like 30 something, and this is only 10 of them, right? Bio 202 is simplifying things down such that you're getting less than one third of all the animal phyla discussed for you, but this is enough. Before we get any further, however, let me clarify a couple of things that I probably need to come clean on. For example, earlier I presented the idea that gastrulation and even the cleavage divisions is something that's more or less universal in all of animals. Now, it doesn't always look exactly the way that I showed you. For example, there's a lot of variation in the kinds of eggs that go into the process of fertilization. You've got some eggs that are really heavy with yolk. Let me draw a picture of this. Okay, so if you sort of visualize the egg like this, right? This is the egg before it gets fertilized. There's always going to be some amount of yolk material, and yolk is made of this material called lecithyl plates, and it tends to settle out. It's more massive, it's really nutrition rich, it's rich with fats and proteins, and what it provides is the nutrition that allows the embryo to develop and grow without relying on any outside source of food. Okay, and so one of the differences that we have among the entire diversity of animals is a lot of variation in the amount of yolk that's there in the egg. You've got microlecithyl eggs. These are ones that have a very small amount of yolk. Then you've got mesolecithyl eggs that have an intermediate amount of yolk. And then the megalecithyl eggs are the ones with a very large quantity of the nutrition. The difference between these kinds of animals basically has to deal with how long the embryo has got to go and grow and develop without an outside source of energy and nutrition. A good example here would be like what we have in the case of mammals. Okay, mammals, a few cell divisions after fertilization, a placental mammal, anyways. I'm talking about placental mammals because we also have other kinds of mammals. In the case of placental mammal, the embryo will start to derive nutrition from the mother circulation via the placenta, and we don't need to rely on the yolk anymore. So for placental mammals, the total amount of yolk that's required to be there in an egg is very little, and those are microlecithyl. In some other kinds of organisms, like in sea urchins, for example, you don't need to have a whole lot of yolk because after gastrulation, the embryo differentiates and it starts to feed as an aquatic larval form. It basically becomes a little zooplankton and it feeds until it's ready to metamorphose into something that looks more like a sea urchin. Okay. Mesolecithyl eggs would include things like frogs, where the egg develops for maybe a little bit longer duration relative to something like a sea urchin or a mammal pre-implantation. But after it reaches a certain point, it becomes a tadpole, a very simple larval-like structure, and then the tadpole is able to feed for itself. Okay? At the opposite end of the spectrum, we've got megalecithyl eggs, which would include things like chickens, which have to grow and develop, and basically they've got to go through a whole lot of changes and growth between the time that the egg is initially laid and incubated, and 22 or 23 days later, a baby chicken, a fully formed baby chicken hatches. All that growth and development has got to happen without any outside nutrition source. And so there, I think it's pretty obvious how a baby chick, developing chick, needs to have a pretty large amount of food in order to make it through that period of growth. So that's one difference. And if you think about something like a chicken, there's no way that the chicken is going to be able to divide this whole massive egg. You're not going to be able to split the egg all the way down the middle like you did in the case of a zygote. Okay, what we drew over here with a zygote being split right down the middle, that's simply not going to happen. What happens is that you end up with um, the egg sort of dividing at the top, but we don't have divisions going all the way down. We end up with a little plate at the top. This is going to be called the blastodisc. And the blastodisc kind of sits at the very top of the egg, and it undergoes gastrulation not all that differently from the way that we described it. Okay. In the case of a mammal, it's a little bit different altogether because you end up with a series of cells around the outside, which are called the trophoblasts. This is kind of like the cells that are responsible for providing the nutrition. And then you've got a relatively small number of cells sitting right on the inside of that. And this is called the blastocyst. And the blastocyst is kind of like the part of the embryo, the part of the mammalian embryo that will undergo gastrulation and form something relatively akin to a gastrula, right? And for something really weird, you've got the insects. When the insects lay this relatively large egg, it's a megalithical egg. It's got a lot of yolk in it. And what happens in the case of an insect egg is that the nucleus, the fertilized nucleus, undergoes a whole bunch of cell divisions. We don't have any nuclear divisions, I mean, and so we don't really have any cell divisions. And then at a certain point, all these nuclei migrate to the outside. And so we end up with an egg with all these nuclei around the outside. And then divisions start forming in between them. We end up with a structure that's got a whole bunch of eggs all the way around the periphery, right? And so this is a different way of developing. But in the end, these are just basically derived forms. These are pretty complex animals, animals that in their past had done things the simple way that we described before with normal gastrulation. But as you're not too surprised to find, there's really a huge amount of variation, a fascinating amount of variation within the animal kingdom. And that's one pretty important thing to take note of. And for the last part of this lecture, we'll be talking about the Hox genes, which requires me to go through a little bit of background. Okay, so Hox genes, we generally think about them as genes that control the activity of other genes. We've talked about these regulatory portions of the genome in the past. Some of them actually code for gene products. Hox genes are among the regulatory regions that actually code for things. And so a Hox gene will produce a Hox protein, and a Hox protein will contain a homeodomain. And the homeodomain is the part of the protein that allows it to interact with DNA. Okay? There are other kinds of proteins that interact with DNA. The homeodomain of the Hox genes are just one class of DNA affecting proteins. Now, the Hox genes we care about in this particular context, and by this context I mean talking about bilateral symmetry, are the ones that specify the anterior to posterior positioning of the body plan of an animal. And sometimes we distinguish these particular Hox genes from all the other genes that are also containing homeodomains and are involved in the development of animals. Sometimes we'll call them Hox cluster genes, which is really quite appropriate because these are largely a series of Hox genes that occur in a linear sequence on a chromosome. So we draw it out like this. And on this chromosome, we've got a tandem array of genes that are all responsible for controlling the development of the fruit fly. And almost all of the genes here are going to be homeobox genes or Hox genes. These genes actually occur in this linear sequence in the same anterior to posterior sequence that you find those regions occurring along the body axis of the animal. So at one end of this cluster, we might call this Hox1. That would correspond to the anterior end of the animal. And at the opposite end, maybe in this case, we've got 8 genes in the sequence. You'd call this Hox8. This would be the posterior end, okay, which is kind of weird. I mean, this was not expected. We learned about this, and it really caught a lot of developmental biologists off guard because we never really anticipated that there would be any need to have the actual genes occurring along the chromosome in the same order, that the body parts occur along the anterior to posterior body axis. For all we knew, for all we suspected, the anterior gene might be in a completely different portion of the genome relative to the posterior gene. Yeah, certainly there would have to be these genes occurring that code for different portions of the animal's body. We didn't expect them to be all there in the same sequence that they occurred along the body axis. And here's where we come to one of the hard and fast rules of animal biology, are the rules of collinearity. And the principal rule of collinearity is that the genes that occur in the Hox cluster occur in more or less the same sequence of body regions along the anterior to posterior body axis. So in the case of the fruit fly, you would basically have this HOX1 gene corresponding to the very front end of the animal. That might be proboscipedia. This gene over here might be corresponding to some area, and behind that, a gene further back might be corresponding to an area of the abdomen, and the very end of the animal is going to be coded for by the HOX8 gene. And this is just one of a few rules that we've learned about since we've become more savvy about how these HOX genes are arranged. Another of these collinearity rules that's pretty easy to kind of visualize is that during development, the deployment of these genes in the HOX cluster occurs in the same chronological sequence that they occur along the anterior to posterior axis. For example, HOX1 is deployed earlier, becomes active earlier than HOX2, and then after HOX2 is deployed, then we get HOX3 and so forth down the axis. So there are a few rules like this, and for you guys, I think just being aware of HOX clusters and the general rules of collinearity is a really good place to start. Now, these rules of collinearity mostly only apply to those animals that have bilateral symmetry. And the reason for this should make a lot of sense, because we talked about this before. A radially symmetrical animal like a sea anemone, remember this guy? Lots of tentacles. Okay. A radially symmetrical animal would have an oral to aboral axis. In the case of another kind of cnidarian, like a jellyfish, you could still have an oral to aboral axis. A jellyfish is kind of like this. You've got a bell on the outside. You got tentacles. You just soft tentacles, and then you've got the bell. You've got this thing called the manubrium. Okay. And then there's the gastrovascular cavity on the inside. And so in the case of a jellyfish, you have the oral to aboral axis, but it's in the opposite direction. Both of these animals are radially symmetrical, but we don't have anything besides the oral aboral axis. We don't have an anterior and a posterior, one end to the other end. And there's something ringing true to the idea that an anterior to posterior axis is something that we'll find in bilateral animals and not necessarily in radially symmetrical animals. Okay? And so, yeah, this is actually true. Bilateria are the animals for which these rules of collinearity apply. Now, it should also not surprise you too much that the number of HOX genes that we find in the genome of an animal, in the HOX cluster of an animal's genome, is in a lot of ways proportional in size. The number of HOX genes is proportional to the relative complexity of the animal's body plan. An animal that's relatively simple, like a flatworm, right? A flatworm where you only have just like a relatively minimal amount of differentiation from the front end to the back, is not going to need to specify as many body regions from the anterior to posterior as compared to something more complex like a fruit fly or a lobster. Okay? And so there's a general rule that simpler animals will have fewer HOX genes, more complex animals will have a greater number of HOX genes. And as long as we're talking about animals with only a single HOX cluster, this is generally true. It doesn't really apply so much to vertebrates because, as you might remember from our earlier discussion, vertebrates actually have 4 separate HOX cluster genes because we've gone through at least a couple of rounds of whole genome duplication. The final piece to our current lecture is going to be that, yeah, not only do we have these rules of collinearity, but also the number of HOX genes is generally proportional to the amount of differentiation that's required in order to specify the overall body complexity of the animal. Okay, that's a good place for me to end this lecture, and I'll pick things up here next time.